12 healthy volunteers agreed to something most studies never attempt. 7 days with nothing but water, no food at all. Every morning, under medical supervision, they gave a blood sample. 2 days before it started, on each of the 7 days, and again 3 days after they began eating. The point was not the fast itself, it was the measurement. Across 117 blood samples, the researchers measured almost 3,000 different proteins. That is about a third of everything currently measurable in plasma, coming from every major organ system. It turns a fast from a story about fuel into a daily record of the entire body. And for 2 days, almost nothing happened. After 24 hours, only 6 proteins had shifted from where they started. After 48 hours, 54. The familiar metabolic switch was running on schedule. Blood sugar falling, fatty acids rising, ketones climbing steadily. But the rest of the body stayed quiet. Then came day 3. From that point, the count grew steeply. By the end of the week, more than 1,000 proteins had changed significantly. 1,044 of them. That is a third of everything measured. The systemic response does not build gradually from hour 1. It waits. This is where it gets practical. The fasting schedules people actually follow are 16 hours, 24 hours, occasionally 48. Every one of them stops before day 3. The authors point out that this fits what the meta-analyses already show. Short intermittent fasting is not superior to simply eating the same calories spread out differently. The changed proteins did not all move together. Sorted by the shape of their trajectory over time, they fell into 9 distinct groups. 3 rose and 6 fell, each on its own clock. Hunger signaling rose early and stayed up. Part of the immune system rose only briefly, around days 2 and 3. Those are the days volunteers describe as the hardest. The strongest theme was not metabolic at all. It was the extracellular matrix, the structural scaffolding between cells found in blood vessel walls and in bone and in the brain. The body appears to start recycling its own structure to spare other protein. The most extreme change of all was in a protein found only in the brain, called tennessine R, which holds together the nets that stabilize synapses. What that means is genuinely unknown. Because weight fell steadily from day 1, while the proteome only shifted from day 3, the two effects could be pulled apart. 452 proteins tracked the fast itself. Only 49 tracked weight. So, whatever a long fast does to the body, most of it is not simply the consequence of getting lighter. Over the week, participants lost 5.7 kilograms. 3.6 of that was lean mass and 1.6 was fat. But 3 days after they started eating again, almost all the lean mass had returned, while the fat loss held. The number on the scale during a fast overstates fat loss considerably. The last part of the study asked what all this might mean for health, and the answer runs both ways. Just over half the links looked protective, the rest did not. Clotting factor 11 rose, and that is genetically tied to a higher risk of clots. This was 12 screened healthy people under medical supervision with no control group. It is a map of what happens, not a recommendation. A 7-day water fast is not something to try on your own. New Age Medica, research translated for everyone.